Hello, everyone. I'm Sahil Bana, and happy you could join us today. I'm one of the sales managers at EAG responsible for our medical device customers. I have a background in analytical chemistry, as well as providing guidance for chemical characterization, testing strategies in the medical device space. We're excited to partner with MCRA on this event. Over the last few years, we've partnered with MCRA in providing valuable insight to help design an effective chemical characterization study, as well as providing consultative feedback for our medical device customers. With the new ISO 10993-1 revision being released, we thought it would be helpful to put together this webinar to discuss the impact on chemical characterization testing, as well as how to navigate your way through a successful study design. We understand the importance of making it through a successful study submission, and the MCRA and EAG team works closely together to help design studies for regulatory submission and help ensure this. I think you'll see we have a passionate team that understands the work they are doing in the lab could ultimately have a major impact on improving or even saving somebody's life. That being said, with today's event, we'll start the presentation from Dr. Soendergaard and Dr. Oberle, followed by a panel discussion with the presenters, as well as Dr. Kevin Trankler with EAG and Dr. Sharlee More from MCRA. The panel discussion will include the questions you sent to us when you registered, as well as some of the poll results that you'll choose from today's events. You could also send your questions live through the chat during the webinar. Again, thanks for joining today, and now I will turn it over to Dr. Soendergaard.
Thank you, Sahil, and thank you, Melissa. Just confirming my slides are up and showing?
Yes.
Perfect. All right. So before we jump into today's presentation, I wanted to just briefly introduce myself. I'm a Director of Regulatory Affairs with Biocompatibility team at MCRA IQVIA. I help clients across a lot of different areas within biocompatibility and biological risk assessment, starting all the way at the early stages of designing your biocompatibility strategy, your biological evaluation plan, troubleshooting protocols, reports, moving that forward to communication with regulators, all the way to the approval process. In the post-market phase, also helping out if questions or issues come up related to biocompatibility once your product hits the market. But today, I wanted to give you a little bit of an introduction to why we're talking about reasonable foreseeable misuse in the context of biological risk assessment, and why it is something you should care about and what it really means. So if you're a biocomp specialist and you operate mostly within ISO 10993 Part 1 in its current version, the concept of reasonable foreseeable misuse might not be something that you're very familiar with or come across very often. And it's quite understandable, because if you go in and look at the current revision of 10993 Part 1, reasonable foreseeable misuse is not mentioned a single time. However, that could be seen as a little bit odd if we look a little bit deeper and look at sort of the context in which we're doing biological risk assessments. So if we look at the greater framework for risk management, which is laid out in ISO 14971 and is further detailed in the companion to that one, TIR 24971, we'll see that reasonable foreseeable misuse is actually a very important part of risk management. It's mentioned 14 times if you do a word search, and it is listed as something that manufacturers shall document as part of their risk management activities, and something that should be identified both during the design phase of your device, but actually throughout the whole life cycle of your product. So with that in mind, you could ask ourselves, like, is reasonable foreseeable misuse something that we should actually already now be looking at as part of our biological risk assessment, and is it a bit of a blind spot or a disconnect between those two standards? So So this idea that there might be a little bit of a misalignment between 14971 and the focus there on reasonably foreseeable misuse and the absence of that in the current revision of 109931 is probably one of the reasons why the upcoming revision that currently is in the final draft stage is now introducing the concept of reasonable foreseeable misuse as part of biological risk assessment as well. Actually, if you kind of do the similar word count in the upcoming revision, assuming that it will go through relatively in the same format that it is in its current draft, it's now featured almost as prominently as it is in the current 14971. So if we assume going forward that reasonable foreseeable misuse will be part of biological risk assessment or 10993 part 1, this really means that all of our biological risk assessments now need to address not only the intended use of our devices, but also any reasonable foreseeable misuse. And the definition that is placed within the new revision leans up against or is pretty much identical to how it is currently stated in 14971. So it is the use of a medical device in a way that is not intended by the manufacturer, but which can result from readily predictable human behavior. And this would include all types of users, so physicians, patients, caregivers, and the types of misuse could be both intentional or unintentional, so intentionally using the device against the eye view, for instance, or unintentionally because there's a risk that you could use it in a way that by accident you did not anticipate. So what it really boils down to is that if we assume that reasonable foreseeable misuse carries forward into the final version of 1093 part 1, which is a good guess at this point, we really need to start thinking about misuse scenarios and how that can change, for instance, the contact type of our device, duration, exposure, and how that translates into how we classify and categorize our medical devices. And that in turn then needs to find its way into our documentation and, for instance, biological evaluation plans, and ultimately in how we conduct risk mitigation. So that says a little bit about why do we need to care about it, and the why is because it is probably coming our way in a very near future, but it doesn't really necessarily address what it is, even though, as said on the previous slide, there is a little bit of a description of what it is. So it is this use of a medical device in a way that you as a manufacturer did not intend, but can be predicted from human behavior, both in intentional and in unintentional ways by all types of users throughout the life cycle. But that's still sort of a fairly broad definition. And while we cannot necessarily predict how regulators ultimately are going to interpret this, or manufacturers how they're going to implement it, I think we can at least take a bit of a guess or make some recommendations about maybe what covers reasonable foreseeable misuse, and maybe also more importantly what it is not. And if we talk about what it is not, really the idea here is not that you sit down and come up and imagine all kinds of wild, unrealistic or extreme misuse scenarios. And it's also not about trying to predict irrational behavior or what one user might do that you never anticipated. So really, this is not about the biocomp specialist or the regulator sitting in isolation and thinking up crazy misuse ideas. So really, we're not talking about oh, is a surgeon going to leave a surgical tool or a sizing guide inside of the patient, and now I have to treat all of my surgical tools as implants. That's really not what we're talking about. And if again we look at the draft of the new revision, it's brought a little bit down to earth again to sort of dispel sort of those extreme scenarios by some of the examples that are listed out. So for instance, it could be, is the device used for longer than anticipated or intended? And that could be, for instance, a device that is intended for limited contact could be used beyond 24 hours. It could be a device that has a different contact categorization. Say, for instance, a device that contacts intact skin. Is there a risk that maybe it could also contact breach surfaces or mucosal membranes? Or would the device likely be used in a patient population outside of the one that you intended? So typical example there would be a device intended for adults that could feasibly be used in a pediatric population. So outside of sort of the scope of what this might mean, it's also equally important to remember that, as I said earlier, it's not about really biocom specialists or regulators sitting independently and coming up with these misuse scenarios. Really, the reasonable foreseeable misuse concept is something that should be baked into both your initial design phase and the life cycle of the device, as we previously talked about. So really, it's more of a team effort and a part of your sort of overall risk management process, and not sort of an event that happens out of context. So really, it's about, when you design your device, you bring a team together and try to figure out, like, are there ways other than our intended use that this device could be used, and is that reasonably foreseeable that misuse would occur? And then as the biocom specialist, the contribution there would be to figure out, like, what would be the impact of this? Is it something that we need to then address either in the design? Are there risk mitigation activities that we should be thinking about? This could include additional testing, it could be labeling, engineering controls, other activities that sort of get ahead of those foreseeable misuse cases. And really, that is sort of a team effort between the entire design team and all the stakeholders in trying to identify those and figure out which ones would fall sort of into the realm of biocompatibility and need to be addressed in your biocompatibility evaluation plan. Similarly, on the post-market side, like biocompatibility already now should be something that you're considering throughout the lifetime of the device, but that also gets expanded to this concept of reasonable foreseeable misuse, where you have to sort of look at what is coming back from the field, what is the real world data telling us about how our device is being used. Is it being used in a way that is no longer aligned with the assumptions we made during the initial design phase? Are some of those unforeseeable misuse cases now coming to light to where you could argue that now it is probably foreseeable misuse? Does that mean that we have to update our biological risk assessment? Are there additional risk mitigation activities that we might have to implement in face of how the device is actually used in the field? Ultimately, this of course puts additional responsibility on the biocom specialist. As a biocompatibility consultant, we fully sort of appreciate that biocompatibility sometimes can be a bit of a transactional activity where a design team hands you a list of materials, an IFU, sort of a general device description, and then you kind of take that and build your biological evaluation plan from that. But in doing it that way, you sort of you're blindsided to some of those foreseeable misuse cases that may have been discussed during the design phase, which didn't make it into the information that was handed to you, but really is now part of the entire biocompatibility risk profile of your device. So the biocomp specialist really needs to be part of all these discussions and kind of bring their expertise and their input to the table, and let the design team know that the design choices that they make have consequences for biocompatibility, and the misuse scenarios that are identified could equally well mean that you would have to conduct additional risk assessment or implement other risk mitigation activities in the early phases. Similarly, again, on the post-market side, the biocomp specialist needs to be in tune with the information coming back from the field. They can't sort of be cut off from that information. So, real-world evidence could inform the biocomp specialist on new, reasonably foreseeable misuse scenarios that, like we said earlier, wasn't anticipated during the initial design phase, but now sort of in light of new information, really would fall into that category of reasonably foreseeable. And again, the biocomp specialist needs to be aware of that so they can alert leadership and the design team that we have potentially new risks that need to be mitigated in some way. So to sum this all up, reasonable foreseeable misuse, while not currently part of 10993 Part 1, I think it's a safe bet to say that this will, in the near future, be part of how we conduct biological risk assessments, so that we're not only considering the intended use of a medical device, but also ways a medical device might be used in a way that was not intended, which could be predicted from how humans normally interact in these scenarios where your device is being used, and that would include both physicians, patients, caregivers, and throughout the life cycle of the device, and that considering these reasonable foreseeable misuse scenarios is really part of the overall risk management process and requires a much more integrated approach in how we do biocompatibility risk assessments and risk mitigations, where biocomp specialists and design teams and stakeholders get together both in the pre-market phase and the post-market phase to make sure that your product stays biocompatible throughout its life cycle, both in the face of how you intend it to be used, but also how it might foreseeably be misused. And with that, I would like to thank you all for your attention. And of course, if you have any questions, if this is something that you're interested in exploring more, feel free to reach out, and happy to help. And with that, I'd like to turn it back to Melissa and Shahil. And again, thank you for joining.
Thank you, Klaus. I'd like to ask our audience to participate in a poll. After I read this poll, a multiple choice question will pop up on your screen. So the situation is, an orthopedic screw is used as a limited duration device during surgery. However, doctors may accidentally lay the screw on the patient while conducting surgery, or may forget to remove the device, and now it is permanently in the body. We are designing a biocompatibility testing plan. The foreseeable misuse scenario is use for longer than period intended by the manufacturer, resulting in longer duration of exposure, and device may be used in an unintended location. And so now I will launch the poll. Now I will turn it over to Dr. Oberle.
All right. Thank you very much, Melissa. Can we confirm that my slides are on the screen appropriately?
Yes, looks great.
Okay, good, thanks. All right, well, thank you for that presentation, Klaus, and thanks for turning control over to me here, Melissa, and thanks, of course, for Shahil for putting this all together. So the topic of this talk today is the role of device misuse in shaping chemical characterization study design and data interpretation. And what I hope to communicate to you all today is, from a device manufacturer perspective, what can you expect from the chemical characterization testing facility in terms of, misuse and how those concepts impact the overall study design. So a couple laundry list items before we start. I'm a relatively new face in the biocompatibility space, so I just wanted to quickly introduce myself. I got my BS in biochemistry at SLU, and then turned around and got my Ph.D. in inorganic chemistry from University of Missouri, St. Louis. From that point, I started working at Eurofins EAG as an analytical chemist, and the majority of my time I've been here about 8 years now, so about 7 years, my expertise was in pharmaceutical deformulation, failure analysis, particle analysis, stuff like that. And I've recently joined the medical device team in support of the chemical characterization of medical devices. So one last laundry list item, just to start off, just because, based on some pre-submitted feedback prior to this webinar, it sounded like there was some interest in the basics of Part 18 study design. So I wanted to have a quick slide on here just so everyone is on the same page from a high level, what goes on in a chemical characterization Part 18 study. So the part that the chemical characterization testing facility plays is basically, when the devices enter in their doors, to the point where the final chemical characterization report comes out. So the devices will come in, and there may have been some feasibility studies or other planning that happened before that. But after the devices come in, they will be extracted according to a protocol that was developed beforehand. And those extracts will go on to a variety of instrumentation, some chromatographic instrumentation, including GC headspace, LC, ICP-MS, and IC, and also some vibrational spectroscopic methods such as FTIR. At which point, after all that data has been collected and interpreted, it goes into a chemical characterization report, which is then, this is kind of when it leaves our door or the testing facility's door, it heads to the toxicologist for a toxicological risk assessment, or sometimes you see it as a TRA. And the information that's provided to them in the report from the testing facility includes estimated concentration of the analytes detected, identification, and confidence level assignment. And that feeds into what the TRA or the toxicologist will use to generate, sort of a margin of safety. So that being said, let's jump back into the topic at hand here, which is the reasonably foreseeable misuse topic. And I wanted to bring this up because, ironically, Claus and we picked some similar quotes out of ISO 14971 and 24971, and I wanted just to highlight some of these lines in section 5.2 if you're interested in pursuing it a little bit more. But it explains here that firstly, of course, the manufacturer shall document the intended use. And that's the easy part for you guys. You generate the IFU. This is what our product is intended for. Now it gets sticky and it gets hard at the second point, where they say the manufacturer shall also document reasonably foreseeable misuse. And it's not until, at least in the current versions, it's not until you get to ISO 24971, in which the ISO method kind of explains what that means. And from the ISO itself, this includes use error, say using a device in the wrong population, intentional acts of misuse, say reusing a single-use medical device, or intentionally using a medical device specified for one operation for another. And they also mentioned that the reasonably foreseeable misuse can be evaluated over the product's life cycle. So that's mentioned in ISO 24971, which is going to come into play a little bit later in the discussion here. But what I found interesting is that they really kind of define everyone's roles in this. So it's kind of the manufacturer, it's up to them to decide what's intended use, what's foreseeable misuse. And then so what the medical device manufacturer is kind of challenged with is balancing worst case scenario in terms of an extraction profile with misuse. So you need to come up with a testing plan or a scenario in which, you know, we're getting the most amount of extractables out of a device while also taking into, you know, into consideration potential misuse. Now, this is really where the testing lab will come into play here. So the medical device manufacturer is really on a quest to, you know, conduct the most suitable device testing for their submission. So you need to find the right lab with strong analytical capabilities, which includes hitting low limits of detection, accurate IGs for that TRA, and swift turnaround so you can keep your submission on target. And you need to find a team, kind of like what Claus was talking about during his talk. It's really a team effort here, who's going to work through these challenging problems with you to address, you know, worst case scenario and misuse, and how we can implement that into the study design to be as protective and as careful as possible for the chemical characterization study to basically feed into that TRA in the back end. At the end of the day, you know, we need to make the regulatory body happy, and it's a team effort. And what the medical device manufacturer should expect is a testing facility that will work with them through these problems. So first steps, you know, everyone likes to say we don't have a crystal ball, which, you know, no one really does. So the first steps you know that should be taken when developing a chemical characterization plan is basically setting the stage early in the program for building around, say, worst case scenario on the front end. So when building or designing a study with worst case scenarios in mind, you know, let's think about the extraction procedure. If your device, you know, doesn't degrade at 50 degrees Celsius, well, let's, by default, out of the gate, let's select 50 degrees Celsius for your extraction parameters. If you have various sizes of a device available, and you're trying to decide which one to send to the testing facility, let's test the largest one. And while the solvent to device ratio should be the same between, say, if you have a small, medium, or large device, what a large device will allow the testing facility is sort of room for a concentration step if that needs to happen. Another thing we can do on the front end before even getting to the doorstep of misuse is, you know, if an IFU suggests the use of a range of a number of devices, say there's a suture and, you know, the IFU says you'll use 3 to 5 sutures, let's test the maximum amount from the get-go just to be as protective as possible. Now that we've set the stage, you know, for what would be worst case scenario, this is where we start building in little pieces for covering misuse. And, you know, one of the lowest lying fruits for this is, you know, what part of the device are we extracting? So, you know, one of the things that could be built into your study parameters is extracting more than just the patient contacting portions. So you'll extract the non-patient contacting portions as well. And this can be useful, especially if you don't have a uniform device. Let's say it's all stainless steel. Say you have a stainless steel probe and then there's a rubber handle or something like that. If we test some of that rubber as well, we can generate an extractables profile that will include some of those components too. So if there was an instance, like Claus was saying earlier, where, you know, a surgeon places, you know, a device down in a patient and they come in bodily contact with the rest of the device, you know, we've you know, the testing facility has already accounted for that, and we have an extractables profile that includes some extra portions of that device. So little things to keep in mind when you're designing your study on the front end. Now, there's other things that we can do in terms of the analytical evaluation threshold. So this is a threshold that the testing facility can play around with during the study design phase to help account for both worst case scenario and misuse. So if the chemical characterization facility sets the AT for a certain value, and that includes all of the worst case scenario parameters. But, the study designer, wants to set that bar a little bit lower to be a little bit more protective, to say, account for longer than intended contact. Well, that's something that your testing facility should be able to accommodate. And that would be, the testing facility would be able to accommodate that by, say, adjusting the DBT, say, moving over to another category of exposure or a longer, term exposure. If you went from temporary to, more permanent, that would drop the DBT, and the AET would drop proportionally. Another thing that can be done on the front end is, again, another example of lowering the AET, and that would be lowering it by adjusting this C parameter here to account for the potential use of multiple devices. So there's a couple of things that can be done in terms of designing what parts of the device are being extracted, to designing how protective we're going to be in terms of identification and chromatography to account for potential misuse. Now, I'm going to come back around to that idea of can be evaluated over the product's life cycle, because there are adjustments that can be made post-study. And one of those, now certainly they're a little bit less desirable than just planning on the front end, but they're certainly feasible. And so what, one of the things I want to point out is there is a possibility that, and as a, study designer looking for the right chemical characterization facility, you should be looking out for a lab that will, be able to or work with you after the fact if adjustments need to be made, say, to the AET on the back end to account for something. Say an unintended, misuse that wasn't foreseen, comes into play. Now we need to go back, reprocess, drop that AET to account for it. The testing facility should be able to go back, reprocess that data, and generate another report to feed to the toxicologist. There's some other reasons that you may drop that AET, and that might be because you're designing a process on the front end to support a future lower AET. Say you're planning on going from temporary use to permanent use. That's stuff that can be pre-baked in there, but there's, that could raise an eyebrow from a regulatory body standpoint if they see the AET, much higher for your temporary use parameters, and then the LOQ, way down here, then, because you have this gap because you're intending to drop it at a certain point for changing the IFU. But that's just something to keep in mind, sort of a separate topic. But this brings up the question: if you're lowering the AET on the back end, and how reliable is that data now? And that is certainly a discussion that should be had with the testing facility. But it's not, say, the end of the world if some case for misuse comes into play and the AET drops proportionally, and our LOQ is now above the AET. That's not sort of the end of the road scenario. We will lose a quantitative analysis perspective, but we can still identify. And with identification information, that can be fed to a toxicologist for the TRA, and those extra things that were identified in dropping the AET, they might be benign or inert or what have you, and it might be justifiable. And so you might be fine with just dropping the AET and reprocessing. Now, certainly, last case, scenario would be, getting a change order, sending in more devices to the testing facility with that lowered AET. So I have just a little, a bit of an interactive graphic here to demonstrate what I meant by that with the AET and the LOQ. So if you look at, the first case scenario where the AET, we're calling it AET1 here, which is just the worst case scenario that includes, worst case scenario extracts, high temperatures, cycles, etc. And that LOQ is set accordingly just below the AET. Now, suddenly, something pops up and we need to drop that AET, now we're considering worst case scenario and misuse. Now all of a sudden our LOQ is above that AET. You know, that's still, you know, we lose some of that quantitative option, but we can semi-quantitate and we can perform and provide, or a testing facility should still be able to provide identification because we're still above the LOD. Now, if some more serious work needs to be had, that LOQ2 can always be applied. And at that point, that would be a situation where the testing facility is getting in more samples or generating more extracts, and they're basically reserving their most sensitive instrument for this even lower LOQ that was set earlier to accommodate this new AET. So adjustments can be made, and in the worst case scenario, with considering misuse, if samples need to be rerun or extract need to be reanalyzed, that should be an option that you could expect from a testing facility. Now, to kind of summarize what we covered for today in my talk, I just wanted to point out that accounting for that reasonably foreseeable misuse is a subjective situation, and it is really sort of a case-by-case scenario thing. But the medical device manufacturer should expect from the testing facility an adaptable analytical strategy that can be worked into the study plan to basically cover as many angles as possible. There are front-end adjustments that we can make in the study design that are more preferable, but there are still back-end adjustments that are still possible if we need to circle back around and reprocess some data and basically account for maybe some misuse or some dropped AET that wasn't not foreseen in the beginning of the study. So with that, I would like to thank you all for your time, and I'd like to thank Claus and the MCRA team for coming along with us for this webinar. I would like to thank Kevin Trankler and the medical device team at EAG, and I'd like to thank you all for your attention. And with that, Melissa, you're clear to take the presentation space back from me. Thank you.
Thank you. Thank you, Carl. Great job. So I'd like to share two more audience polls. So in this first poll, a brain shunt is designed to be used on adult patients. However, the design is appropriately small and may be used on neonate population. We have already conducted biocompatibility testing, including chemical characterization and a toxicological risk assessment. The foreseeable misuse scenario is use in a different population, including vulnerable population, than intended by the manufacturer. And so now I will launch the poll. For our final poll, the wound dressing instructions for use state do not use more than once per day for 2 weeks. However, medical professionals are conducting frequent dressing changes, and these wound dressings may be used for over 30 days. We are designing a biocompatibility testing plan. The foreseeable misuse scenario will be considered a long-term exposure compared to prolonged exposure. And so now I will launch the polls. I want to thank you for participating in the polls. And so now I will turn it over to Dr. Bana for our panel discussion.
Thank you, Melissa, and thank you, Claus and Carl, for your insightful presentations. Now our presenters and panelists will join us for the panel discussion. We had a number of great questions, so I'm gonna have to pick and choose here, but let's start with this one. The biggest hurdle we face in our biocomp testing is providing all the devices for the testing. will designing chemical characterization studies for device misuse scenarios lead to me needing to give more devices? EAG team, MCRA team, feel free to.
Question. I think it could. A lot of times it will lead to more devices. Kind of to the point that Carl was trying to make in his presentation, there can be some creative ways to, really kind of minimize the amount of devices that we need, but I think it's really around risk and risk management. So I think it's very case specific, but yes, in some cases it will require additional devices. I don't know if the MCRA team has any thoughts on that either. But.
I think it is kind of how you said, you need to evaluate, is it an actual feasible scenario, or is it something that, again, we're just kind of playing a little bit wishy-washy? Or you can also see if there's ways that you can handle on your TRA later.
Yeah, so I mean, I think it can be managed around on the chemistry side, especially if there's adequate room between the AET and the LOQ, and we can drive that AET down without getting too close to the limit of quantitation. Then the devices that you would normally use in a study would be sufficient. Pretty case specific. It's a good question.
Thanks, guys.
I think the only thing that I would add is just echoing what Kevin said there about, more devices is preferable. Concentration is always an alternative, but concentration steps always kind of, they tend to raise a red flag. Now, certainly a chemical characterization testing facility will provide spike and recovery data to support a concentration step if that was the route that we took. But always, I think if you can send more samples, that's the way to do it.
All right, thanks, guys. On to the next question. How do I implement this into my devices that I already conducted biocomp on? Do I need to do anything else for my already marketed devices?
Well, I think this is one of those that are going to be a little bit unknown until we maybe see how regulators are interpreting things. I mean, the 1093 Part 1, in the current version, upcoming version, kind of has some language to the effect that that the issuance of the new standard doesn't necessarily mandate any new testing for a device that is already on the market. So there's not necessarily an expectation for sort of a retrospective application of biocompatibility assessment for a device that you already have on the market. But I think there might be sort of a gray zone if later on you're resubmitting either it could be for EU MDR, you're resubmitting your technical files, or if you have a PMA supplement, if you have a 510K submission for what would otherwise be maybe a lesser risk. It is possible that regulators might sort of re-adjudicate your biocompatibility at that point, where they get it in front of them a little bit more fresh. But yeah, I don't know that we know yet exactly how this will be applied to devices that are already on the market.
Thanks, Klaus. Anyone else have anything to add to that?
I mean, I guess if I would add anything, it would be, there's the potential of leveraging your chemical characterization data to support that, that this new evaluation with foreseeable misuse has minimal impact. As far as the broad biocompatibility, I think, I defer to what Klaus said for sure. The question is, can you go back and look at the data like Carl suggested, and can they go deeper? Can that be used to leverage? Can that be leveraged to support that your device is safe? It's a good question.
Yeah, I think another. Oh, go ahead, Sharlee.
Oh, go ahead, Carl, and then I can go.
Okay. Yeah, well, this is just another sort of comment on the chemical characterization side. This is not something that I have encountered directly, but I can imagine that if testing facility A performed the first chemical characterization, and then there was to be some scrutiny on that device that demanded the AET get dropped and reprocess the data and provide another a report for a new toxicological risk assessment. I don't know what that would look like if you took that data and you sent it to testing facility B to take testing facility A's data. So I think that you would want to at least go back to the original testing facility, and if they weren't available anymore, if that team was not available anymore, it may result in retesting at that point.
I think I was just going to quickly bring up that you can always go check your BRA and see whether you were considering foreseeable misuse. And if it wasn't, go check with the design team and see was this included and whether it really is something you need to start thinking about. And if you do, you can reach out to your teams to start doing it and figuring out, are your MOSs going to be fine? Are we talking about how drastic of changes are we talking about with these scenarios?
Okay, thanks, guys. Next question. Do you think a pre-sub and getting FDA alignment is a good first step? Or will I be inviting the reviewer to come up with unique scenarios? That's an interesting question.
I think that's for Claus or Sharlee.
I guess I can start. I think it goes. I see the underlying question is like, am I just inviting the reviewer or FDA to start coming up with crazy scenarios like we talked about, and really it goes back to like that's not really the intention here. The defining reasonable foreseeable misuse is really on the manufacturer. It's not on the regulator to come up with these scenarios. So if it is something that you should do, so I don't think there's any reason why not to bring it up, but I wouldn't necessarily shine a light on it and sort of uninvited send a list of potential misuse scenarios to FDA and say, can you please help me pick the ones you think are the most relevant one, because they might say all of them. On the other hand, going forward, if there is an expectation that you address reasonable foreseeable misuse, I mean, you need to be fair and honest about like, what did we sort of uncover in the design phase? Like, is there something where we can say, this is probably going to be evident to FDA and us as well. Here's our plan for how to address it. Give some very specific, examples of what you intend to do. So, for instance, saying that we anticipate that this device may be used for more than 24 hours. Therefore, we will do these additional risk mitigation activities, which could be testing, which could be other things. Does the FDA consider this to reasonably address this unforeseeable or this foreseeable misuse scenario? Don't come in with open-ended questions. Be very specific about it, or else you are going to risk sort of getting a laundry list of requests if you're not being proactive on it. And I'd say that really goes for a lot of your biocompatibility evaluation plan. If you communicated that to regulators, you're the expert on your device. You should be informing them on like what is the outcome of your risk assessment, what is reasonable in your opinion. Here's what I'm going to do, and then the question is, like, does FDA or does a notified body, whoever you're talking to, do you see anything that is not covered here, and not sort of ask them, can you please mention other things that I should do? So that would be my advice.
Thanks, Klaus. Sharlee, anything to add? Okay. Next question. Why do I have to adjust my AET or perform more chemical characterization? Can't my toxicologist just evaluate the exposure for 2 devices or a chronic exposure, period? It's probably also directed to the MCRA team.
Well, I think I can answer this one. I'm sure Klaus can as well, probably Sharlee too. The problem with that is you will be missing, there will be potential analytes you're missing. So the AET is driven by the AET calculation, which includes the number of devices, and the number of devices is inversely proportional to the AET. So the more devices that your patient's exposed to, the lower that AET will be. And so if you assume 1 device, but it's really 2, anything below that AET will not get picked up and analyzed and will not even be shared with the toxicologist. So at that point, you can't accommodate that extra exposure. So it needs to be done at the chemical characterization stage. Sharlee, do you disagree?
Nope, that's exactly it. I can only do my tox risk assessment on what I know, and I don't know what now could be there if you have, and above this limit, if you have 2 devices being implanted. So.
One last quick question. This is more of a, I think, going to be an opinionated answer, but will -1 lead to a reduced or increased requirement for ENL testing?
Can of worms question.
I think it could initially lead to more testing because there will be situations that people need to revisit, or there'll be confusion or misalignments about what is reasonably foreseeable misuse. But once we settle into a pattern, I think it'll be pretty neutral and remain about the same.
Yeah.
Opinion.
I think. Oh, yeah, go ahead, Carl.
I was just going to say that there's a couple of changes that I think are coming up that could introduce some more testing on the in the early term, and that's we'll have to wait and see what how regulatory bodies respond. But doing end of life testing, that's something coming, and the chemical characterization testing facilities are would be prepared to do that, and that would require additional testing. Another change that I think is coming down the pipe is changing the exposure times. I think is coming down, where anything say over 5 minutes is being counted 1 day. I might have that a little bit mixed up, but I think the exposure times are getting exaggerated a little bit, and that could involve adjustments to the AET and recalculation.
Okay, we got a few minutes left, so I did want to also go over one of the poll scenario discussions. So, it'd be nice to talk to you through the wound dressing poll. So I kind of want to direct this to you, Klaus. How do you feel about the poll results? What first steps would you take?
Well, I mean, this is one of those that I think we've seen a few times where there's a little bit of a strategy of labeling your way out of like those long-term endpoints. I think while you could say let's do some risk mitigation and do some labeling, and I see that's the second option there. That's already what's kind of happening and kind of what's wrong with some of these IFUs is that they'll say used for up to 30 days, and then we all know very well that they're being used longer than that. So yeah, the design your ChemCare and TRA to address long-term use is probably the right way to go. I think you're gonna have increasingly difficulty explaining the actual real-world use with an IFU that says don't use for more than 2 weeks or 30 days. So I would also pick design a study to account for longer term exposure.
Yeah. And then I guess to the EAG team, how difficult do you think that would be to implement that misuse into the actual chemical characterization study?
I mean, for this one, I don't think it would be difficult, especially if it's planned for on the front end. You know, going from prolonged to long-term exposure, you know, you're dropping down the AT by an order of magnitude. I think it would be more difficult in that scenario if you're trying to go back to your analytical data and address the risk around it. I think if you design it on the front end, I think you're in a good spot here.
Okay. All right. Well, thank you guys for your answers, and thanks to the audience for joining. It's been a pleasure to be a part of the event today. If you want to delve deeper into any of this stuff, feel free to contact us, and we're here to help you guys. And yeah, thank you. Melissa, I'll leave it with you.
All right. Thank you to our presenters and panelists, and thank you to all our attendees. All of you will receive an email with a recording of this webinar, and we'll also share the other poll results with you and additional resources. If you have any other questions, please email Sahil at the address on your screen, sahil.bana@meeurofinsus.com, or even easier, eag.com/contact. Once you leave today's webinar, you'll receive a survey on the presentation, and we would really appreciate your feedback. And so on behalf of EAG and MCRA, thank you so much for joining us, and have a great day.